Dear students, today I am going to discuss about SDS PAGE, sodium dodecyl sulfate polyacrylamide gel electrophoresis. What is SDS PAGE? SDS PAGE is a bioseparation technique. Okay, so this analytical technique was first developed by Ulrich K. Lamb. So this technique is actually helps proteins or nucleic acids to separate based on their molecular weight. SDS is a detergent which with strong protein denaturing effect, and it also binds with the protein and imparts negative charges. Okay, and along with SDS we will also use DTT dithreotretol and beta mercaptoethanol. These are the reducing agents which breaks the disulfide bonds, and this is actually critical for the protein folding and the protein structure. So in the presence of SDS and the reducing agent, the proteins will unfold and it will form the linear structure, that is the polypeptide chain with the negative charge imparted on it. Okay, now how it helps in the separation based on their molecular weight. So here we will be using a gel. Okay, the polyacrylamide gel. The name stands, that is, it is a polymer of acrylamide. Okay, now here I will discuss how to do this, how to perform this SDS page in the lab. For that, we have to do stock preparation, preparation of gel, sample preparation, electrophoresis, then fixation, staining, destaining, and observation. Okay, so step by step, we will be discussing on this. This is the gel stacking or the left side image is for the gel and the right side image is the electrophoresis apparatus. Okay. So for gel stock, we'll be needing to prepare acrylamide 30% stock and two different trays. So the trace, one trace solution with 8.8 pH and another trace solution with pH 6.8, and sodium dodecyl sulfate 10% stock. Along with that, we have to prepare ammonium persulfate 10% stock, but this stock we can only use a freshly prepared stock. Okay, so that means you can keep it for 2-3 days in the 4-degree fridge, but not more than that. Okay, and timid, it's a tetraethyl methyl ethylene diamine. So this compound is a liquid which will help in the acrylamide polymerization process. Okay, and also double distilled water. Now for the electrophoresis part, we will be needing running buffer, lamelli buffer, staining solution, running buffer and lamelli buffer. These two for electrophoresis. Okay, and after the electrophoresis is completed, we have to use fixative, staining solution, and the distiller. Okay. So three part basically. First part is the gel casting. Then second part is electrophoresis. And before electrophoresis, we also need to do the sample preparation. Okay. And then comes the staining. Okay. So first part, that is the preparation of gel. How to do that? So in this process of gel preparation, that is polymerization of acrylamide, we have to be very careful about the pH of the trace buffer as well as about the freshly prepared cross-linking agent, that is ammonium persulfate, and also timid. Timid is a light sensitive agent and also it is evaporable. So keep your timid stock very nicely. Now here we have to make some cautions. You can see the cautions are written over here, that you have to use a freshly prepared APS, and also as soon as we add the timid, this is very critical, that we can prepare the trace, acrylamide, everything we can just add, but as soon as you add the timid, the cross-linking will start. So we have to immediately transfer that solution to the gel slab. Okay, and then we will have to layer the isopropanol on the on top of the resolving gel. So suppose you make the glass plate assembly and then you pour the resolving gel and you forgot to add the isopropanol, then what will happen? Air oxidation of the acrylamide will happen. So to prevent that, you have to do that. And after the polymerization is done, then you can wash off the isopropanol with distilled water. After that, we'll add the stacking gel where the acrylamide percentage is less. So I forgot to tell you that in resolving gel, the acrylamide percentage will be more. So it will be more dense, more compact gel where actually the separation will happen. Okay. Now the stacking part, here the 5% acrylamide is there. And here the cathode side is on the top and anode side is on the lower side of the gel, because SDS page is a vertical electrophoresis. Okay, so here if your proteins are masked or marked with negative charges, then it will be easily migrating to the anode side. If we are not doing that, then it will be, I mean, if we are not using SDS, if we are not using DTT or beta marker-propanol, then it will be a native page. Okay, but when we are using SDS, that means it is a denaturing page. We are going to observe the molecular weight of the protein when it is a polypeptide chain. So the sample preparation, obviously, we have to use the lamellae buffer here, which is also known as a cracking dye. So in the cracking dye or in the lamellae buffer, we have trace, we have SDS, and a little amount of glycerol, bromophenol blue as a cracking dye, and DTT or beta-methylpropionol. So that means 1:1 ratio. We have to mix our sample and the lamellae buffer. Then we have to put it in a dry bath or a water bath and like boil for 2-3 minutes. Okay, so our sample is done, our gel is done. So now we can go ahead with the electrophoresis. And in the electrophoresis, we have to keep in mind that during the stacking portion, when the protein is passing, we can set the voltage 90 volt, and then we can increase the voltage to between, we can keep it in between 150 to 200 volts. And we'll do the running of the electrophoresis till the tracking dye starts coming out. Okay, so you can vary this little more or less. So between 30 to 40 minutes, your resolving should run the electrophoresis. Now in the electrophoresis buffer, trace glycine SDS, this is the common composition. After that, we have to separate the glass slabs from the gel, and this is the gel which is in the water. You can see the blue gel in the petri dish, and this is after the imaging, not the same gel obviously. So this is an example of a overexpression of a protein. So before induction and after induction, so you can see this dense band which is overexpressed. So the observation will be only obtained when you finish the staining of the gel and the destaining. So here you can see the all the blue background, but here the blue background is minimized because of destaining. Okay, thank you very much.